Has the nucleus of a differentiated cell undergone an irreversible change? Does it still have all the genetic information to produce all the differentiated cells in the body? And if so, can it still express all that information? Robert Briggs and Thomas King studied these questions. For their studies on frog embryos, they needed to develop 3 techniques. They needed to remove the nuclei from the host egg, enucleating them. They needed to isolate the nuclei from donor cells, and they needed to transfer the donor nuclei into the host egg. Others had shown that an unfertilized egg can be activated for development by simply pricking it with a glass needle. Briggs and King then isolated nuclei from cells in frog embryos at the blastula stage of development. Then they inserted the nuclei into activated, enucleated host eggs. In 60% of their trials, the eggs with their new nuclei developed into normal swimming tadpoles. The fates of blastula cells are not yet determined. What if one takes nuclei from more advanced embryos? As Briggs and King took nuclei from later and later embryos, late blastula, early gastrula, late gastrula, neurula, and early tadpoles with tail buds, fewer and fewer of the transplanted nuclei led to normal embryonic development. In the frog embryo, nuclear potency does decrease as development proceeds.